Good afternoon, and welcome to UCF Coastal's Lunch on the Coast speaker series. I'm Dr. Graham Worthy, Director of UCF Coastal. Through this speaker series, I hope you gain appreciation for the extent of basic and applied coastal research underway here at UCF. Orlando may be an inland city, but we still contribute to the issues facing our coast, and are in turn impacted by those same issues. One of the goals of UCF Coastal is to grow public awareness about the amazing coastal resources we frequently take for granted. To achieve that goal, we've assembled an incredible team of cutting-edge researchers who serve as a resource for everyone from marine scientists to the general public, businesses, and policymakers. Our Lunch on the Coast speaker series is running from July through December, with one speaker each month. Through this series, we're proud to highlight some of the recent research undertaken by the diverse interdisciplinary faculty of UCF Coastal. You'll learn what it really means to live in a coastal state, with expert perspectives ranging from archaeology to coastal planning, from engineering and economics to ecosystem and human health. You'll get an appreciation for the full range of issues currently facing our coasts and the resources that UCF Coastal is bringing to bear to help foster an understanding of those threats and hopefully chart a path forward. Now let me pass you over to our moderator, who will be introducing you to this month's speaker, and we'll also be running our question and answer session following the talk. Thank you for joining us today, and we hope you enjoy the Lunch on the Coast speaker series.
Hi, my name is Kyle Martin. I'm the Director of Content Development for the College of Sciences. I will be acting as your moderator today. Today, we are excited to kick off the Lunch on the Coast speaker series. This is a very exciting collection of experts that we have assembled. Today, we are starting with Dr. Melanie Beasley from the Department of Chemistry. Dr. Beasley's research includes the fate and transport of contaminants in our environment. She studies both terrestrial and marine systems in order to understand how contaminants affect not only the biogeochemistry of an ecosystem, but also the microbial ecology. Exactly what that means will be explained in the video starting right now.
So we'll let that run for a bit. So we'll let that run for a bit.
Hi, my name is Melanie Beasley, and in my lab we study contamination, whether that contamination is in surface waters, freshwater, marine waters, or even our wastewaters. In order to solve a problem, you have to know that it's actually there. And so in order to do that, we have to take samples. We go out into the field, we take water samples, soil sediments, sediment samples, soil samples, and whatever the contaminant of concern is, we extract and then we measure to see how concentrated that is in our environment. Over the last year, since the COVID-19 pandemic occurred, the University of Central Florida has been very concerned about how we can manage that here on campus. One of the many ways that the university does that is looking at our wastewater, because we know that we can see the SARS-CoV-2 virus in wastewater waste. Or we have, over the past spring semester, set out samplers at certain dorms where we can collect the wastewater over a 24-hour composite, bring it back to the lab, and then test it for the SARS-CoV-2 virus. So in essence, an overall theme of our lab is that we study contamination, whether it's nutrients or some type of a chemical, maybe even a particular pathogen, a bacteria. But what we're studying it is in the matrix that we collect it in.
All right, so now I'd like to introduce Dr. Beasley, who will be giving our talk today. I will remind you that there is a Q&A section at the bottom down here. If you have any questions during the talk, feel free to type those in. You can also upvote a question if somebody's question matches your own. And all right, we'll give it over to Dr. Beasley.
Thank you so much, Kyle. I appreciate that. And I am so excited to be
the first Lunch on the Coast seminar series today. And I got to tell you, ever since I was a young child, the coast and the oceans has been a fascination to me. And as a young graduate student in chemical oceanography, ever since then, about 20 years ago, I have made looking and understanding our waters around us one of the primary areas of my research. And so I'd like to share with you today some of the work that we've been doing more recently, and it involves microbes. So when I first started out, I was studying chemistry. I was a chemist major. I understood all about chemistry in the oceans and in our environment. However, it was partway through graduate school where I realized and I started learning that microbes are a major part of what goes on in our environment. And let's talk about that. So what I invite you to do today, I want you to think about your everyday life. Think about when you get up in the morning, you go through your day, you go to sleep at night. Can you think of places and times and instances where microbes have a major influence on what you're doing today? All right, let's think about that. So let's talk about what is a microbe. Well, a microbe, just by its essence, by its name, micro, means that it's very, very small. Typically, they're primarily microscopic, free-living organisms, most of them. There are some that you could see with the naked eye, but most of them are microscopic. They are things that we hear about called viruses. They are bacteria, archaea, even some eukaryotes like protozoa, as well as maybe some microscopic algae. And so these microbes are very, very tiny. If you look at this period right here on the screen, if you think about the bacteria that many of us know about, E. coli, it would take 10 to the 9th cells of E. coli to fit in this one little spot right here. What is 10 to the 9th? That's a really big number, right? That's actually 1 billion. It takes 1 billion cells of E. coli just to fit in here. And so if we think about our everyday life and how microbes affect us, can you think about that? Well, one of the first things I said that a microbe is a virus, right? Well, nowadays, viruses, the COVID virus, is very much a part of our life, right? It has become something that is ever-reaching around the world and how we are affected in not only our everyday lives, but in our health, right? Also, when you think about bacteria, maybe you think about, maybe you've had a strep throat, or maybe you've had other kind of illness where you've had to have an antibiotic prescribed to get rid of that bacteria in your system. So a lot of people, when they think about microbes, they think about it from a negative connotation, right? We think about how bad they are. But if I were to tell you that, would you be surprised if I told you that only 1 in 1 billion bacteria or microbes that are in the world are actually pathogenic to us? There's only really a few that actually harm us. The rest, and there's trillion of them or more, that actually are being very beneficial, or maybe they're very just harmless to us. But there's a lot of things we don't know about them and what they're doing in nature. There's a lot of fascinating things that we do know. And so those are some of the things that I like to read about and I like to research. So let's talk about other things, other ways in which microbes affect your life. Can you think about any other ways? Well, when it comes to the environment, one of the things that I think about is something that we're doing right now. Every second of our day, what are we doing? We're breathing, right? And what are we breathing in? We're breathing oxygen. Where do you think that oxygen came from? When Earth first formed over, what, billions of years ago, there was no oxygen in our atmosphere. So where did that oxygen come from? Well, about 2 billion years ago, there were these microscopic little algae that lived in the ocean, and they were called cyanobacteria. And all of a sudden, what they started doing, well, not all of a sudden, they evolved to be able to take CO2, carbon dioxide, and convert it into O2, into oxygen, using a very well-known mechanism that we all know is photosynthesis. And so over 2 billion years, these little tiny microbes in the ocean, they just constantly started churning out all this oxygen, right? And so today, how much oxygen is in our environment? If you've ever learned about the environment or the atmosphere, we have 21% of oxygen, and that's the only way that we are able to breathe, to metabolize, to live as we are today. And it's all because of these tiny, tiny little microscopic algae that live all over the ocean. And would it surprise you to know that these guys are still pumping out oxygen, and they pump out 80% of all the oxygen that is in our atmosphere at any time? Much more than the trees and the plants that are on the terrestrial side. So these guys, we need to take care of them, right? We need to make sure that they keep producing all that oxygen. Can you think of any other ways that microbes affect your everyday life? Can you think about, maybe you've heard the term microbiome. Well, microbiome is a term that we've kind of heard about over the last 10 years or so. It's become more popular. And maybe, what does that mean? Well, biome means a habitat, a particular habitat where a group of somethings live. Well, a microbiome is a habitat in which a group of microorganisms live. Maybe you've heard the term gut microbiome. Well, what's gut microbiome? Well, that is all the different bacteria that live in our stomach, in our intestines. And what do you think they're doing? Every time we eat, they are processing all the food that we eat. They're breaking it down. They're producing all these great macromolecules like glucose that are allowing us to be healthy. If we have some bad gut microbiome, maybe we're not quite as healthy as we should be. But if we have good, that makes us healthy. And they provide a lot of great benefits to us just every time we eat. And so let's think about other areas in which we might have microbes in our systems. There is microbiomes on our skin. We also have them all within every part of our bodies. And so one of the cool things about microbes is that they communicate with each other. And you know how they do that? They do that by releasing these compounds, these organic compounds. And anybody that's taken organic chemistry or been in a lab of organic chemistry knows that a lot of organic compounds have a smell, right? Some of them smell good, some of them don't smell so great. Some of them are kind of whatever. But anyway, when you have these microbes that are producing these smells, well, maybe that's why we have to wear deodorant every day, right? Or can you, and this is another thing that when I was reading about what some of the really cool microbes do, you know what, one of the things that I thought was really kind of fun, one of the things that they do is they kind of help our love life. Would you know that microbes, they release all these different odors and smells and scents, and they contribute to how we smell every day. And so a lot of times that has to do with how we attract the people that we love. And so sometimes if you think about it, if they are producing all of this sense about us, and what have we been doing this last year? We've been using hand sanitizers like crazy. And so if hand sanitizers and cleaning all of our surface kill 99.9% of our germs, oh my goodness, what are we doing to our microbiome? And are they affecting our love lives? Well, that's something to think about, isn't it? It's a lot of fun. So anyway, let's go back to our microbes, all right? And let's talk about how they are in the environment, the things that I like to study. So let's talk about what the environment is. All right, so for me, the environment includes the oceans. Think about the oceans, you have the water in the oceans, you have all the sediment that's at the bottom of the oceans, and then you also have your coastal waters where you have your beaches, you also have your wetlands, and maybe some of your marshes, which are really cool ecosystems for bacteria as well as other organisms, as well as we have our marshes and our wetlands. We have a lot of those here in Florida, right? And then we also think about our surface waters. Think about those lakes that might be in your backyard or down the street, the rivers and our streams. We also, for me, the environment not only is that water, but also the sediment that is down below. And then also there is groundwater. Groundwater flows beneath our feet in the soils, carrying constituents with it. And so those are also areas that I like to study. And of late, we've even started studying wastewater. And so we'll get into that in a little bit, because wastewater, every time, think about what is wastewater. Every time you turn on your sink in your home or at work, every time you flush your toilet, every time you wash some clothes, all of that goes down the sink, goes down the drain, and it goes into your waste. And so if you live in a house that has a septic system, it goes into your septic tank. And in that septic tank are a whole slew of bacteria that are going to town on that waste. It might not have a septic system. You might have piping that takes it to the wastewater treatment plant, but that same thing happens there. All of that waste is broken down by bacteria and other microbes. And so those are all of these areas, or the environment, that I like to study. And so here we have, let's take a look at Orange County. This is the map of Orange County. We see where UCF is right over here, and Orlando is, the downtown is right here. What do you see that is really cool about this area in Central Florida? Well, look at all these areas. What is all that? Blue. It's all water, all this water. And you see all of these streams and lakes and tributaries. And what's really unique about it is they're all interconnected, aren't they? Here's the St. Johns River. And that's what's very interesting about water. No matter where it is on land, it all sometimes eventually ends up in the oceans. And so that's something that's very cool, right? So we have to take care of our water, no matter where it is. We have to be careful with what we do with and how we contaminate it, okay? So one of the things that's very exciting to me is how microbes and their relationship with contaminants. So I talked about contaminants before in that little vignette. So what do I mean by contaminants? Well, over the years, I've studied a whole slew of different contaminants, whether they be some heavy metals. Maybe you've heard of lead in drinking water. That's a problem. For a long time, I studied uranium and how uranium contaminated soils. Also, other heavy metals might be a contaminant that we've studied. Also, organic molecules, organic compounds, hydrocarbons. What are hydrocarbons? Well, hydrocarbons are the components of what you find in crude oil. So for many, many years, especially over the last 10 years, I've been studying how hydrocarbons in the Gulf of Mexico have been processed and degraded after the Deepwater Horizon oil spill. So it was a large, big project that we worked on, and how our organisms can break those down. Microbes can take hydrocarbons and degrade them all, use those carbon as a carbon source and degrade them all the way down to even CO2. And so they are one of the major players and one of the major avenues for degrading contaminants, especially organic contaminants in our environment. That's what we call natural attenuation. It's where the microbes that live there degrade the contaminants that are there. Also, some of the emerging contaminants that we study are things like sucralose and maybe personal care products, things like gadolinium that are MRI contrast agents. These are called emerging because they've been there in our environment, but we're just now starting to realize they might be a problem. But one of the things I'm going to concentrate on today is nutrients. Well, you think, how can nutrients be a contaminant, right? Well, what are nutrients? Things like nitrogen and phosphorus. Every living human being or every living organism needs nitrogen and phosphorus to live. Those are our primary nutrients. However, if you have too much of them, what can happen? Well, you can have problems. And what do we have in Florida, in our waterways, that is a result of too many nutrients? Well, we have algal blooms, right? And those are not necessarily great, especially if they go too long, they can degrade our water quality. And so today, what I want to talk about are biocontaminants and how we can use these organisms that live in these systems to help track and understand these biocontaminants. Okay, so let's talk about biocontaminants to waters. What is exactly a biocontaminant? Well, to me, a biocontaminant is what we would call waste going into water that contributes to the nitrogen and phosphorus. So when we eliminate waste as humans, the waste that we eliminate has a lot of nitrogen and phosphorus in it. Our septic systems, if you have a septic tank that's leaking, it's leaking into the groundwater around your system, and then eventually it might be leaking into your local lake or your local river, and that might be contributing the excess nutrients to that system, and then maybe a result or leading to the result of algal blooms. Also, our stormwater, we have a lot of things like not necessarily biocontaminants, but it might move the biocontaminants. You can also have fertilizer runoff and so forth, but also our domesticated animals. If you're going out and you're walking your dog and you don't clean up after him, well, that particular waste can eventually go into your local waters and lead to a contamination problem. Birds, we have lots of birds. That's one of the things I love about Florida. You can't go anywhere without maybe seeing a heron or maybe one of those great sandhill cranes that you have to stop your car while they're walking across the street, right? And then of course, our farm animals, they have a different kind of a metabolism, and there are a different type of way that they process something called a rumen in the cow, and that's how they process their waste. And so we understand that these biocontaminants going into water can affect our waterways. So let's say, for instance, you're going to your local water facility or your local water lake or wherever that you like to swim at. Well, if it's got a lot of fecal material and a lot of bio waste in it, you don't really want to go there, right? So it is the responsibility of a lot of the regulators and the regulatory agencies in our areas to monitor the waste as well, or the nutrients going into our waterways, to see if there's ways that we can control it and to fix it. And so a typical way in which methods for detecting biocontaminants in water, the primary way is what we call fecal coliform testing. Well, this is really just a way of seeing if you have fecal bacteria in your waters. It doesn't tell you where it's coming from. Well, in our lab, we have started working with, or over the last several years, we have been using these new methods, which is using DNA testing, which is called microbial source tracking. And so it's a way to track biocontaminants in water, but being more specific, being able to say, okay, that particular contaminant is coming from humans, or that particular contaminant is coming from a dog. And so this is some of the things that we're working on in the lab. And we've been working on it for the last 4 years here in Orange County. And so the way that we do this is that we know, remember back to our gut microbiome, remember those bacteria that are going in there? Well, everybody has specific to different, the humans have a different type of system, gut, the way that their metabolism works, the way that the bacteria are in there, that they have a very different type of, some of the bacteria that are in their system are different, say from dogs, different from birds, different from other birds. And so what we do is we find bacteria that are very specific that we all as humans have. And so we all have the Bacteroides dorii here in our gut. And so that is one way we can track that humans are, there is a human contaminant in our waters. Back to our canine friends have a very specific type of bacteria that lives in their gut, as does our bird friends as well. And so if we test these waters, we can say, instead of just using the very broad fecal coliform method, we can say, hey, we've got more human waste in this water, biocontamination, or more dog or more bird. And then maybe we can find better ways in which to mitigate that or fix that problem. Also, if you've got a lake that has a very specific bird problem, like one that I worked on here in Orange County that had a very poor problem with cormorants. Cormorants are these really interesting birds. You might have seen them. They're black like this, and they stand there with their wings out because they're drying them. And so they roost at a very specific lake, or they roost around a lot of our lakes. And so this one lake had a very specific fecal coliform problem, and so a high nutrient problem. And so they were wanting to know, well, is the cormorant one of the main problems with that? And that's one of the things that we studied. And so instead of looking just at very specific bacteria that are just for all birds, well, we developed methods in which we could look specifically for these cormorants. And so one of the ways that, or the way that we test this, and then we go to the lab, is we use a very common thing called a PCR. Many of you probably know all about it. And we can use quantitative PCR. And this is a way where we have standards, we develop standard curves, and we can do analytical quantification, just like you would any other analytical technique. We can do it with our DNA. And so we use this using primers and probes that are very specific to those particular bacteria we were looking for, or that particular bird. And so over the years, we've been doing this for about 4 to 5 years now in Orange County, and there are some very specific overall trends that we're seeing. We see these human signatures in a lot of our lakes, but we don't really see it too much in our streams and our rivers, because maybe the streams and rivers, they might be moving faster, the water might not be building up these contaminants as easy to say that a lake that still moves. Many of our lakes are actually connected to each other, and they move water through each other as they go down toward the ocean, but they might not move as fast. And so we also see that we see more of a human signature in lakes that are in residential areas, so where you might see a lot of septic tank use. And then also, which was really interesting, is last summer in 2020, we saw a bigger increase in the amount of human signature in our lakes than we had in the years before. And so I got to think about that. How could that be? Well, one of the reasons possibly is, what were we all doing last summer? Well, we were all sitting at home, working from home, right? Well, we weren't going to work using the facilities at work. We were at home using our own facilities 24/7. So that might be one of the reasons why we see this higher increase of a human signature in our waters last summer. Also with dogs, we see a positive canine signature, primarily in lakes and not so much in the streams and rivers. We also see it in parks and recreational areas, especially where we see dog parks where people congregate and maybe walk their dogs. And also, again, we saw an increase last summer, and it could possibly be because people were at home. Plus, a lot of people, from what we know, adopted pets during the COVID time. And so maybe that's the reason why we're seeing a higher signal of the dogs and of dog signals in our waters last summer. And then also our birds. There is no specific trend for birds. Birds are everywhere. They're ubiquitous here in Central Florida. We don't see a difference between lakes and streams. They are there, of course, in some areas a little bit larger than others, but there's no correlation between residential and industrial areas. Then back to our cormorants, the specific cormorants, we do find them in a lake where there is specific cormorant roosting. What's interesting, we actually find it in the sediment. So that DNA, we know that the cormorants have been contributing to that lake for many, many years if it's in the sediment. And then we also see these higher concentrations during the times that the cormorants are there roosting, which is in the fall and the winter. And so this is what we've been doing to help, hopefully, with our local regulatory agencies like EPA and DEP, and helping them find specific ways in which they can help mitigate some of these biocontaminants in our waterways. Now, I want to switch over just a little bit to our work that we've been doing here on the UCF campus, where we've been studying the SARS-CoV-2 in wastewater. And this is something we call wastewater-based epidemiology. This is something that's been used for many, many years. For years, even before COVID, it was used to track polio in certain areas, as well as other viruses, hepatitis, and so forth. And so when the COVID-2, or when the COVID-19 pandemic started, you started seeing an increase in people studying how we can use wastewater to track the COVID signal. And what we found out was that when a person is sick with COVID, whether they know it or not, whether they're symptomatic or not, excuse me, they shed it in their waste. And so we're able to actually see that virus capsid in the waste. Not see it because it's microscopic, right? But we can measure for it. And so we know that a person who has COVID will shed the virus in their waste up immediately when they start. And so they'll be shedding it even before they might know they're sick, or before they're even symptomatic if they are. And so even if they're not symptomatic, it will be shedded in their waste. And so we can actually see it possibly almost several days to a week before they actually know they have it. And so it can almost be like an early warning signal to help us determine if somebody, but with wastewater, you can't specifically find one person that has it. You're looking at a whole community. And so many of you have probably seen the SARS-CoV-2, this little guy here. This is the virus. He's a little bit of a larger virus. He's got a larger genome. He's about 30,000 base pairs in length with his genome. And maybe you've heard about these spike proteins. These are these little things that look like spikes coming out. It makes them sticky, so they'll stick to things and can move about. And then it has RNA inside of it. And that's what we're looking for. We're looking for that RNA because it will tell us how much of that virus we have. And so the reason it's called corona is because it looks very similar to the sun's corona here. And so that's the reason why it's been named the coronavirus. And so when we decided that UCF, well, it wasn't we, UCF decided that when the COVID pandemic started last year, UCF was looking for ways in which they could monitor it on campus and control it on campus. And so one of the things they did were they were doing individual testing for people, of students as well as people on campus. They were also doing contract tracing. And one of the ways in which there was a big push to start doing wastewater testing, because there was all over the country and all over the world, we saw this huge influx of this wastewater testing being done. It got to the point where it was hard to even start, because getting the pumps that we need, like you see here, was hard because everybody was wanting to do it. And so everything was on back order, right? And so UCF wanted to do this so that we could look at it, say one building. So if you have a dorm, can you find track or monitor if COVID is in that, if people in that dorm have COVID, and not specifically, you won't be able to say this person or that person has it, but as a whole, do you see an increase? And so what we're looking for is a signal to say, okay, is it increasing in this particular area? And if it is increasing, well, maybe we need to get these people tested and take a look at maybe doing some contact tracing. And so that's how we started. So back in the late fall, we started working for this in our lab, and we started developing the methods. We went out and got the equipment we needed. We have the pumps. And what these pumps do, if you think about where the wastewater is, well, the wastewater is down usually about 15 to 20 feet below in a manhole. And so you have these pumps that have to pump up. You put the tubing down into the manhole and you pump it up. Well, what these pumps do is they are composite pumps. And so you can program them to where they'll bring up a certain amount of wastewater every, say, hour or something. So you can do it over a 24-hour composite. So you can get a nice sample over that time frame. And so we got our pumps. A next thing was trying to find the best place to put them. On UCF, if you see here the map, this is the map of UCF, and all these little these yellow dots are manholes. And this is where wastewater travels underground throughout our campus. And so if you notice, they're all kind of connected. And so some of these weren't for specific dorms, they were for a lot of dorms. So we would need to find something that was specific to, say, 1 or 2 dorms. Also, some of these manholes were in the middle of the street, so we couldn't necessarily put them there. And so it was a bit of a challenge to find the best place to put them. And then also with the great help of our UCF utilities, the folks there, we found the best places, we got them installed. You might have seen them around campus this past semester. And so we got those going. You see how they go into the manhole and then they pull up, and we collect water from all of these places, wastewater from all these places each week during the semester. And one of the things that is important when we are processing the wastewater, well, if you think about wastewater, number one, it has COVID in it possibly, but it also has a lot of other pathogens. So you had to be very, very careful. And with the help of our great people at EHS, they helped us develop methods in the lab for being safe. There has never been an instance of where you've gotten COVID from your wastewater or from your waste. However, there's other pathogens, like I said, so we want to be really careful. And so here are the great people in my lab. Here's Leah and Genesis and Matt, and they helped us develop this method and get it going. And you see here some of the factors that we had to come across when we were working with wastewater. Well, how much volume do we use? Do we use all of this right here, or do we process this whole waste? Do we do just liter, 500 milliliters? Those are the important things we had to make some decisions about. And there's another thing, it's what I call my goop factor. Well, wastewater can be a bit goopy. Some of it can be a little less goopy, some can be more goopy. And so that goop factor has a lot to do with how hard it is to process that waste at first, right? And so we also had to have the necessary lab equipment so that we can be careful using things like a biological safety cabinet that keeps any kind of pathogens inside it and not into the air within our lab. Also, we had to be safe using all of our great PPE here, which is our personal protection equipment, the gowns, the masks, and of course the shields. And then we also had to develop our specific protocols of how we were going to be safe with this. And we also had to be cognizant of how we were going to, the waste that was left over, how are we going to sterilize it? Because you can't just put something down the drain, right? It has to be sterilized. So we go through a lot of bleach in our lab. All right, so the next part is you have your wastewater sample. Well, what are you going to do next? Well, you have to pre-concentrate it. So let's say we take about a liter of water and we pre-concentrate it down to either using a filtering technique, which is what we did for most of the semester. The spring semester, you put your wastewater here, using the vacuum filtration of your lab, it pulls the water through it, and any of the virus or any of your waste or your DNA, your RNA is stuck right there on that filter. And then we take the filter, and that's what we extract the RNA from. Now, at this time, a lot of places around the country, not just universities, but also municipalities were testing their wastewater, and we started seeing publications coming out about the methods people were using. It was like the Wild West. Everybody was using a different method. And so it still really has not gotten down to the point where we have an established method for doing this. So we figured out the method that worked best for us and how we were able to do it. But there's other methods. There's things like filtering like we did. You can do ultrafiltration. That's kind of hard to do though when you have a really goopy wastewater. Ultracentrifugation, you can do that, but you have to have those really great high speed ultracentrifuges, and I did not have one, so we weren't using that. Another method that we've recently started is salting and flocculation. It's an old molecular biological technique using something like skim milk, powdered skim milk, which flocculates the virus out. And so we've recently started using that. And then after you pre-concentrate your water, you then extract your RNA. Remember, all that RNA is in that capsid. And so we extract the RNA. And when you work with RNA, you have to be very, very careful because there's RNases out there that love to eat up your RNA. So you have to be very careful, especially for cleaning your surfaces, using your biological safety cabinet. tons of bleach and ethanol and RNase removal solutions. Now, in the past, I primarily worked with DNA, and that's a little bit easier. It's not quite as sensitive to degradation. It can be, but not as bad as RNA. So we had to develop specific methods in the lab to be very careful with the RNA, because that's what we were pulling out for the COVID. All right, so if we're going to detect it, we're going to use the same methods that we did for our microbial source tracking just a little bit ago. That was what was really great about it, because we'd already been doing this for years in the lab. We now can transition over using the RNA and trying it for and working with it with the COVID, and so, or with the SARS-CoV-2. And so what was really nice is that the CDC had already developed these primers and probes that were used for being able to detect the SARS-CoV-2 virus in our water. So we did not have to design those, which was really nice. And so for us, we were using the N1 and the N2 genes. Those were from the they came from regions in the nucleocapsid. And then we also have these positive controls that we were able to get from IDT. And so that is how we were able to establish our standard curves, and then our develop the method for determining the SARS-CoV-2 in the wastewater. And so we were very successful. We were able to see it. Here is a graph of just from the spring semester. And so we see that at the beginning of the semester, in our dorms, or the dorms we were testing, that we had a slight signature in the background, and then it kind of went away. But right about the time we saw spring breaks around Florida, we started seeing upticks. And then you remember we had our spring break a little bit later than usual. And so the week of spring break, it was at 0. And then all of a sudden, right after spring break, we did see a big bump. And so this was kind of what we were hoping we would not see, but of course that is what we did see. And so after that, it did level out. Of course, this is at the end of the semester. Now, these were from the dorms that we were testing. At this time, we are continuing, we have kind of transitioned here in the summertime to not look at specifically at the dorms, but we're going to be looking at larger areas of campus. And so you remember this map that I showed you, where all these little manholes, if you walk along the street, I bet you if you look down, about every so often you're going to see a manhole, right? Well, there are other areas, if you notice, where all these arrows go to. There are areas on campus called lift stations, and there's different lift stations all over campus that collect some of the water that is in a different area of campus, and then it transfers it and then shoots it off to the wastewater treatment plant down the street. All right, so we have transitioned our samplers to 5 of these lift stations, and so we're able to cover all of campus. So we'll have a lift station, we'll sample from a lift station that covers this area, one that covers this area, one that covers the area in the middle where we have a lot of our classrooms and our dorms, our classroom buildings and our buildings, and then we'll have one down here in the south, and then we have a master lift station where we study that collects basically all of the wastewater from all over campus. And the reason we're doing this is that now that we've gotten into this time of higher vaccinations, we're not going to be testing as much. If on campus, we have about 10,000 students who live on our dorms, but every day during a normal semester, we have 10 to 20,000 more students and faculty and staff that come on our campuses. And so there's a lot of unknowns going into our fall semester. We're going to be full, we're already back full time. And so there's a lot of unknowns coming as far as the SARS-CoV-2 and how we can monitor it and keep us safe on campus this fall. And so what this will do, it'll allow us to be able to track it over a larger area so that we can see how well our vaccinations are working, how well we are maintaining and monitoring, or how we are keeping the COVID signature down here on campus. And so that's some of the things that we're interested to know, because we really don't know going forward how things are going to go, right? There's still a lot of unknowns. And so one of the very interesting things, there's an interesting Twitter account that has a database of all the different places around the country and around the world that are doing wastewater testing. And this is the wastewater epidemiology has the testing for that has just blossomed over this last year. So many people, all of these are different universities. The blue are the universities, and you have other entities like wastewater facilities and so forth. Here in Florida, there's several as well, including us. We're right here. And so one of the things that we are considering, that we want to consider when we're talking about this wastewater epidemiology, is that it's been used for many years. We've known this, and it's been used to track other things. But we were able to successfully do it here on campus and actually be able to show and give us some idea and be able to have an idea of how well how well we are able to keep that the spread of the virus down. And so one of the things that the COVID-19 pandemic has highlighted is the relevance of this wastewater-based epidemiology. People are doing it all over with success, meaning that you're able to see. And what's really interesting, some of the larger towns and cities around the country, like Boston and Cleveland, started doing this last year early on. And what is very interesting is if you look at the COVID signal over there for the SARS-CoV-2 and their wastewater over those time periods, it follows very closely, very, very closely with the clinical numbers of people who were sick. And so it, in essence, it can be a little bit of an early warning device, but it also can tell you a lot about your community-based, from a community-based contagion. And so what's interesting now, if we've established this infrastructure, we have the pumps in place, can we use this, be prepared later on if we have other future pandemics that happen? We're already better placed with going further and applying this technique in the future. And hopefully it'll also give us some ideas going into the fall of what's happening, because once you're vaccinated, we're going to be doing less testing. And so this will give us a better idea of how COVID might be on our campus and where it is. All right. So with that, I want to thank you all for being and listening. And I can go on and on about all this. I'm always excited about my research, but I got to stop now. And I want to thank all the people in my lab who have been just instrumental in getting this off the ground. Leah and Genesis here, they were excellent students they had just graduated, and they came and they helped me get this going over the last 7 months. They have since left me. Genesis has gone on to medical school and Leah is going to grad school, but now we have Matthew Blow, who came in and has taken over. And we also have my great graduate students, Anthony and Nicholas Young, who have been very instrumental in keeping, Nick has been helping with the wastewater with this area as well. Both of them have, and so I want to thank them, as well as our undergrad students who come in and help with some of the cleaning and maybe some of the boring stuff. And I appreciate all of them so much. And this is a picture when I was thinking about this talk and what I wanted to talk about. I found this from my early years. It's from 20 years ago. And it shows that this was in the Houston ship channel. We were doing some studies, and you see I was climbing around in the mud back then. I still am today. And then also I want to have a big shout out to the friends of the Beasley lab who made essential contributions for when we were developing the MST project and the canine side of it. So they were very essential to helping us develop our method. And so I want to thank all of them. And then also our funding is the University of Central Florida. They've been very instrumental in helping us getting the wastewater COVID tracking study off the ground. Also for our MST studies, Orange County and the city of Orlando. Our president, UCF, Dr. Cartwright, he has been very instrumental in supporting our wastewater testing and has been an integral part of that, as well as Rhonda Bishop and Renee Michelle at EHS, Dr. Dyken and Jackie Hopp at Health Services, UCF Health Services have also been very helpful. David Bach and Don Sibley at EHS were very helpful at the very beginning in getting our protocols down and also finding where we're going to put our pumps. And then also all the guys at the UCF facilities who help us pop those manhole covers and find the best places for the wastewater testing. Dale Lance, Mark Mayhew, and Riley Baird. I really want to thank them as well. So with that, I thank you all, and I will be happy to answer any questions. Thank you so much.
All right, thank you, Dr. Beasley. It's been an interesting lunch conversation having you. I
Oh,
appreciate
I can't go not talk when you talk about, I've always said, is that a lunch conversation? But I know it's part of our what we do every day, right? So
Very much is. All right, I'm going to go off this question list over here. So how have biocontaminants contributed to the seagrass die-off and manatee starvation in the Indian River Lagoon?
oh my goodness. Wow. That's a great question. I'm not, and I got to say, I am not familiar. I don't do that type of research and I don't understand about, all the intricacies of that. But it's interesting. There's been some recent talk about, maybe there is some, from the manatee side, anytime you have microbes are going to have a major impact on any ecological system. They're going to have positive and they might have negative. And so I really can't answer that because I don't really know that. I can make some guesses, but I don't want to do that.
That's fine. Where do the maybe I don't know, this may be a plumbing question for you, but where do the contents of a septic tank go? Like how does
where do they go?
If there's a leak or if there's some other way to where's the contamination coming from septic tanks?
Well, if you have a septic tank, now I'm not a septic tank expert, but what I do know about it is your waste, that anything you flush down your toilet goes into your septic tank or anything down your drains as well. And even think about your washing machine, the water from your washing machine and the soap goes down into that as well. All right, so there are specific bacteria that live in your septic system that are there breaking all of that down, all of that waste. And so it just keeps breaking down. If it's working correctly, it's breaking down. If you don't have any leaks, you probably, from what I understand, probably have to have it kind of cleaned out every 10 years, I think is what, but I'm not an expert on that. Don't quote me on that. But a lot of these septic tanks might be old. I was really surprised when I came to Florida how many septic systems are even in downtown Orlando. I have a septic system at my house. A lot of septic tank systems are along the coast. And so a lot of them, they break down. And just like any other type of a tank, they will leak. And so when they leak, they will leak out any of that waste and that, the water from that waste, and it might have pathogens in it. It probably has a lot of nutrients in it, a lot of nitrogen and phosphorus. And so once it gets into the soil around your septic tank, it will then start moving with your groundwater, and then it can eventually get to the coast or to any kind of water body. And so it can contribute to, and it has been seen to contribute to some of our algal blooms. It can be a contributor to those.
Can you correlate the volume of COVID found versus the number of infections? So I guess probably related to campus and the
they say that there's been some back of the envelope calculations. I think there's a couple of papers where they have done some calculations like that. Usually, typically, it's in the larger sewer sheds where you have maybe 1 million people, and so it's a larger statistical measurement. And you can, and at these places like the water utilities, where they have instances of the amount of flow that's coming through and the amount of the population of where the waste is coming from. So there has been some of those. However, I haven't really tried to do that because it really wouldn't matter. It really wouldn't make, I really couldn't make any contribution to that, other than the fact that typically I know with the contact tracing and with what we know about how the student health center is, we can have maybe just 2 or maybe 3 people in a dorm, depending on how many people live in that dorm, and we can find that signature in the wastewater. We can't find that specific person. We don't want to do that, but we can find the signal.
Okay. When we talk about wastewater in broad terms, is COVID-19 shed only in feces or also in urine?
I don't think they found it in urine. I think it's primarily in the feces.
Okay, so just waste.
It typically comes from this. Oh, I know this is getting for lunch. The swallowing of the mucus, because it affects you here. And I don't know if they've actually found if it's enteric, where it's it recirculates or it's working in your stomach. I think it's primarily coming from the mucus, or if that's what they think.
Here's a interesting question. So do you think we may ever shift to personal testing for COVID-19 using fecal samples instead of addition to the nasal swab?
I think they do. I think there's probably already some tests out there that do. because it depends on. But I think if you go into a place to get tested, would you rather? Where would you rather be tested? You see what I'm saying? So
guys already had their brain
ease, the ease. And so there's other tests out there where they've studied your breath, also other, different ways of testing. Yes.
What factors make it easy or difficult to detect a contaminant using existing cost-effective methods? I guess, or maybe put another way, what are some of the challenges of using existing methods for testing?
Well, if are we talking about COVID, or do you think that
detecting contaminants? So maybe in a broad sense.
Contaminants, in a broad sense. Well, it really and that's really my more of my expertise is when you come up with contaminant. If you're thinking about a chemical contaminant, it really depends on what it is, because what it is how you go about determining its method of detection. So if it's a heavy metal, you want to use something called an ICP-MS, which is an inductively coupled plasma mass spectrometer, and then you want to, depending on if it's in water or if it's in sediment, you want to process your sample knowing and extracting that metal. However, if it's an organic compound like our sucralose we've been testing recently, or if it's like a hydrocarbon, you process that a whole different way because it's an organic compound, and you also test it differently. You use different instrumentation like HPLC, high performance liquid chromatography, or mass spec as well, LC mass spec. So it just depends on what that type of contaminant is and where it's at, because a lot of times it makes it very challenging is if you're looking for a contaminant that is in low concentration and it's either in your water or it's in your mud, and that sediment holds on to it. So you have to be able to figure out ways in which you can extract it successfully from that system. And that is a lot of times the most challenging part in how you extract it from the environment, just to get it to where you can have enough of it to be able to test for it.
Let's see, what do you know? So regarding the COVID-19 virus, do you know what is the efficacy and survivability of the virus in the wastewater?
That's always been a question that we've had, because the way that when we're doing our filtering, we're assuming that it's still in the capsid, okay? Because the way that we filter it, the virus is smaller than our filter pore size. And so what we end up doing is we acidify our sample to the point where everything is protonated, and meaning that it has hydrogens on it, and that it has no negative, it has a positive charge, okay? And then our filter has a negative charge. And so you're hopefully collecting it. Now, me personally, knowing how goopy these samples are, a lot of times I think it's more of a physical, not just that electrostatic keeping it on the filter. It's just everything, those filters get clogged up really quickly. And so it just gets stuck there. Now, we don't know for a fact how much of it is the survivable virus versus the RNA that comes out. We do know that if it's in the RNA form, it's not going to last very long. But if it's in that COVID, if it's in the capsid form, it's going to be much more resistant to degradation, though we do have to process it as quickly as possible. Heat will degrade it. So when we collect these samples, we process them right away. And so otherwise, we also keep them on ice and cold. So by just the fact that we're finding it in the wastewater, that's telling me it's surviving it to an extent. However, we don't really know how well it's surviving it are we catching all of it? Or are we just catching a small portion of it? That's always going to be a question.
All right. Can WBE be used to detect any other public health epidemics that are not pathogen-based? So for example, malnutrition or diabetes?
I don't know. That's a good question. How would you, I'm not familiar with what the body does when it has diabetes. If you release, I know you release certain things in your urine that can be tested. I don't know. Honestly, I'm not sure about that, about things that are non-pathogenic. We can test for things like STIs and other viruses as well, But that's a good question. I'm going to have to look that up to see if, you know, if you can track like things like diabetes. I'm not really sure. That's a great question.
That's the beauty of science, you know, it's always exploration,
Yes,
finding the next
that
answer,
is.
something else.
I promise you, I had never worked with a virus before last year, and so this has been very interesting.
All right, I think this will be our last question. Can similar methods be used to study microbes that are early warning systems for events like climate change or climate events?
Oh, sure. Oh, yeah, they're being done right now. There are some really cool bacteria out there. Oh my goodness, there are bacteria that produce electricity. There are bacteria that can survive and live in highly radioactive places. If you think about climate change, also, one of the things, and I didn't really talk about it today, that I'm very interested in is the community of bacteria, what we call microbial ecology. And let's say you have a marsh system and it's on the coast, and if it's a very healthy system, you have a high diversity of bacteria that live there, you have other organisms that live there, everybody's happy. But then let's say all of a sudden you have an oil spill, or let's say all of a sudden you have some kind of catastrophic event that's affecting that soil or that sediment or that coastal system. What's going to happen is all the bacteria that live there that can't handle that stress are going to die off. And then those that can handle it are going to be very opportunistic and take over. And so some of those might not necessarily be really great bacteria that we want to take over. And so in essence, you're going to have the diversity of that ecological system go down because a lot of the bacteria, a lot of microbes are going to die off. Same thing with think about what happens. It depends on what kind of process or what type of things we're talking about with climate change. With climate change, you can have areas where you have salinity intrusion, where the coastal areas are the salt water is intruding into fresh water. That's definitely going to have a major effect on the type of bacteria that live in that system. And you can monitor the community of bacteria, and it also might kind of be a bit of an early warning device of telling you if you have a little bit of a contamination that's starting, and you can see that change in that community, but you have to be monitoring it. Also, heat is a big difference with microbes. What's really interesting about microbes, they live in every ecological niche in our environment. There are some microbes that can survive and live in 130 degrees C temperature waters that are above boiling. There are some that live in the deep ice of the Antarctic, very cold. They live in high salinity, low salinity, high pH, low pH. So if you have climate change that is affecting either temperature, salinity, pH, you're going to see a change in that microbial community depending on how they can tolerate that stress. And so, yes, there are many, many studies out there that are using microbes to help us detect climate change and the different changes that is occurring. That's a great question.
Great. All right. Well, we'll give a virtual round of applause for Dr. Beasley. Thank you for coming out today and sharing your expertise on this. We hope you enjoyed the presentation. I would encourage our audience, if you could join us again for our next Lunch on the Coast on Tuesday, August 10th. Again at 12 p.m. It's called What? No Oysters for Lunch. It's going to be presented by Stacy Barber and the Department of Anthropology. So this concludes our webinar. Thank you so much.
Thank you.